Okay, in this screencast we're going to have a look at linked genes, and to start off we'll have a look at something that's a bit familiar. We'll have a look at a pair of homologous chromosomes, and we'll have a look at a dihybrid relationship of these genes. So again, hopefully we're familiar with how this actually works, and we can imagine in this position on a particular chromosome we have the locus of a particular gene, and we can have the allele, which could be big A. Or on the other chromosome we could have the allele, which is small a, and we can imagine, you know, this being the number 3 chromosome and this being the number 7 chromosome. So you've got two different chromosomes. The other allele that we might have might be on this number 7 chromosome, and we could have big B or small b just working. So what this is really showing us is we've got two alleles, the A allele, which we're heterozygous for, the B allele, which we're heterozygous for, and that these are located on separate chromosomes. Let's have a look how this works with linked genes. And links basically referring to the alleles being on the same chromosome. So in this example here, we might have big A and big B on the same chromosome, and we might have little a and little b on the same chromosome. So essentially it's the same. We have an individual with a big A, and they also have a small a, they have a big B, and they have a small b as well. So they're heterozygous for A and heterozygous for B. Now, when we write this, we write a little bit differently. We can draw it, sort of looks a bit like a fraction with one over the other. What's this? This is showing us is that this A and B are on one chromosome, and the small a and b, the recessive alleles, are on the other chromosome. And you can imagine this over here. The big A and B on this side here are always going to be inherited together on the same chromosome. The little a and the little b, the recessive alleles, are always going to be inherited together. Okay, they're going to travel together. Okay. Let's just look at how we write these symbols. First off, we'll look at the normal dihybrid relationship, and when we write it with a quite often, like we see above, sometimes we write it with a little semicolon in between them, and that semicolon is really probably the more accurate way of writing it. It's showing that they exist on separate chromosomes. If we see it typed when they're, this is the linked example here. If we see it typed, often we see it written with this sort of slash on the angle there. And the other way we can see it written is how I showed you just previously, with one over the other. And you know, I really like to think of it as this being one chromosome and the line separating the other chromosome. So almost imagining that we've got, you know, one chromosome like this with one lot, and one chromosome, you know, out there with the other lot, and it's just showing that they're attached and together. Okay, so let's have a look at how this might work in a particular cross. Let's cross some parents. Let's go with some individuals here that are heterozygous for both A and B. Okay, and I'm using my writing here to show that the individuals are heterozygous. They've got both the dominant and recessive alleles, but I'm also showing that the genes are linked by drawing in this particular way. The gametes that could be produced, well, because the A and B hang together, big A, big B, the two dominant alleles, is one possible gamete. The other possible gamete is the two recessive alleles. Same on the other side. Alright. And when we write these into our Punnett square, we write them down the side, gametes down the side, offspring in the middle. It actually becomes quite a simple Punnett square compared to what we're used to. okay, and our offspring, again, writing them using symbols to show that the genes are linked. They don't suddenly become unlinked just because we've done a cross here. And we're going to see we have some individuals here. The relationship and the ratios that we get are going to be exactly the same as what our monohybrid crosses are. This individual here is going to be recessive for both traits. These two are going to be heterozygous, and this individual here is going to be homozygous dominant for both. When we punch it into our genotype and phenotype ratios, you're going to end up with that phenotypic ratio of 3:1. Okay. Okay, let's pause things for a second there in the middle of our problem and have a quick look at crossing over. Now, crossing over, we talked about in meiosis, and really the importance of it we'll see right now. The situation we had before, we had genes which we said were linked, these being two homologous chromosomes. We said big A and big B always occurred on the same chromosome, and we said on the other situation a small a and a small b, that being the locus of the B alleles, this being the locus of the A alleles. So if crossing over occurs, we might not have that happen. We might not have big A, the dominant big A, always being inherited with the dominant big B. Now let's just have a look at how that happens after crossing over and sections of the chromosomes swap, that crossing over point being the chiasma. Let's have a look at the outcomes on our alleles down here. So we can imagine a big A sitting up here. All of a sudden, a big B can be swapped over to the other chromosome, and up here we have little b with a little a. We can now have some alternative combinations. Now the likelihood of those combinations we might be able to make out on this fuzzy diagram here a little bit. It's on this diagram, it's showing a number of different alleles or loci of different genes on a pair of homologous chromosomes. Now we'd expect very little crossing over between A and B. They're really close together. There's not much room for the chromosomes actually to cross over. So if A and B were inherited to get, if they were linked and we were discussing them, we'd expect them to be linked nearly all the time. The other example here is if we looked at A and a chromosome down here, let's look at K, okay? And if we looked at A and K, there's a whole lot of spots where crossing over might occur between them. So we'd probably expect that crossing over to be a lot more likely. So what this means is that as the locus of the alleles becomes further apart, crossing over is a lot more likely. When the locus is close together, it's a lot more likely they just present themselves as linked genes and they're inherited together. Okay, let's have a look at how crossing over might affect our example that we worked before. Look, if crossing over was to occur, what we could have is that this big A here could mix with our small b here, or the small b here could also be inherited with the big B. So you could have different combinations, which would give us combinations like these gametes here. Now the same thing could happen over this side here, and the likelihood of these happening, like we talked about before, really relies on how close together those alleles are on the chromosomes. So as we write them in, we could see it starts to look a lot more like a dihybrid cross, and in fact it basically becomes it. The only difference is all the alleles that we have being the genotypes that are being produced all the way down here and all the way across here, the likelihood of them is a lot less likely than the linked genes. The linked genes are what we'd expect nearly all the time. The frequency of these will really depend on how close together the alleles are on the chromosome.